today's topic of discussion is applications of bioinformatics. Now, in this picture, you can see that bioinformatics has widespread applications. It has application in different fields of biology as well as chemistry, as well as pharmaceutical technology. So it has application in medical informatics, computational biology, pharmacogenetics, immunoinformatics, proteomics, genomics, pharmacogenomics, and cheminformatics. So in all these fields, there is an application of bioinformatics databases as well as computational tools. So why do we use bioinformatics? Now these are the reasons for which we are using bioinformatic databases and tools. First application is storage and retrieval of biological information. So with the help of sequencing machines, automated DNA sequencers or next generation sequencing platforms, we are generating this genomic DNA sequences. We are determining genomic DNA sequences of various organisms, right? We are determining the protein sequences, protein structures. So where we save this data, there must be some kind of storage system available for the storage of this data, and there this databases, biological databases come into picture. These biological databases, they are available free of cost, and you can submit your data into these databases. So once you determine the sequence, once you determine the structure, it is not necessary to you just store that data on your local computer, but instead of that you just submit that data to the database, and it can be accessible by anybody in the scientific community worldwide. Moreover, you can retrieve the existing stored data on the database. So some other researchers who have submitted the data into database, that can be retrieved by you or anybody. That is a very basic application of bioinformatics: storage of the biological information and retrieval of that information, or storage of the data and retrieval of the data. And for this purpose, we use databases. Then retrieve or compare gene sequences. You can download the gene sequence from the database and you can compare that gene sequence with the other gene sequences. Okay. Then predict function of unknown genes or proteins. So many times what happen, researcher determine a gene sequence or researcher determine the protein sequence, but he or she does not know the function of that gene or function of that protein. So we can predict the putative function, tentative function of that genes and proteins by using bioinformatic tools. Then search for previously known function of the gene. So suppose a gene sequence is present in the database along with its function, then you can search for a previously known function of that gene. Then you can compare your own data with the other researchers' data. Suppose you determine the insulin protein sequence or insulin gene sequence. Now you want to see whether there are any errors or not, or is there any mutation or not. So what you can do, you can compare your data with the other researchers, because other researchers must have also determined those sequences and submitted in the database. So you can simply compare your sequence that has been determined by you with the sequences that have been determined by other researchers. And moreover, you can compile the data and distribute the data to other researchers. So once you submit your data into database, it will be available to everyone in the scientific community, free of cost. So in this way, with the help of database, you can distribute the data to other researchers so that they can do the further work in that field. So these are some basic reasons for the uses of bioinformatics. Now, bioinformatics found application in variety of fields. I will explain these fields or I will explain these applications one by one. We will take some examples also. So, knowledge-based drug designing. Now, what is knowledge-based drug designing? All of you know that these pharmaceutical companies, they have this R&D wing or R&D division, that is research and development division, where new drugs are discovered and these new drugs undergo extensive clinical trials, and if the FDA, that is Food and Drug Administration, the regulatory authority, gives the approval to that drug, then that drug will be marketed by the company. So in the traditional approach or the conventional approach of the drug discovery, these companies, they have this chemical compound libraries where the synthesized chemical compounds are stored. Now the library size may range from 1 lakh compound to almost around 10 lakh compounds. Now these chemicals either have been synthesized by that company by themselves, or they can procure that library from the library provider. So suppose there is another company who has the library, so they can provide that chemical compound library to the other company who is doing R&D, and for that purpose they will charge something. But the point is that these libraries contain maybe millions of compound, 1 million compound or 0.1 million compounds, and you have to figure out of this uh, million compound, which compounds is showing this pharmaceutical activity, or which compound is able to inhibit that pathogen or kill that pathogen for which you are finding out a drug. So first of all, you identify uh, you identify the disease and you identify the target, and for that target, in order to inhibit that target or in order to kill that target, you have to screen this entire chemical compound library. So what you will do the laboratory experiment in which you will see you will see that which compound is able to show that pharmaceutical activity, right? So in order to do this is called as a bioassay. This is called as a bioassay. Now, in order to do this kind of bioassay, high throughput screening methods are used, where robotic arms are used to perform this bioassay. Okay, so in the high throughput screening, all these millions of compounds are screened, and after screening, you will come to know that which compounds are showing the proper activity against that pathogen or against that disease. So during this process, what will happen? A lot of money will be spent. So it's a costly process, and a lot of time will be spent because you have to do this bioassay experimentally in the laboratory. So that leads to increase in the cost of the drug discovery. So in a survey, it has been found that any drug which has been starting from its discovery phase to the market phase, it takes around 10 to 12 years, and it costs more than 1 billion US dollars. मतलब एक दवाई डिस्कवरी फेज से मार्केट तक पहुंचने में लगभग ऑन एन एवरेज 10 to 12 ईयर का पीरियड लगता है, और जो कॉस्ट है उसके लिए ऑन एन एवरेज इट इज 1 बिलियन यूएस डॉलर्स. ओके, बिकॉज वंस यू डिस्कवर द ड्रग, जब भी आप ड्रग डिस्कवर करते हो, यू फाइंड आउट दैट दैट ड्रग इज हैविंग दैट potency to either kill that pathogen or to cure that disease, then you have to undergo the clinical trials. The drug has to undergo the clinical trials. Now in clinical trial, we test for safety and efficacy of that drug. Whether the drug is safe means, uh, if it has a side effect, then it is of no use. So we test for its safety, whether it has any toxic effect in the patient's body or not, that we test. And we also test its efficacy, means its efficiency to cure the disease, right? Now, if the drug has more efficacy and if the drug has no side effect, then FDA or regulatory authority will give the approval for the marketing of that drug. Okay, so this period from the discovery phase to the marketing phase, I'm talking about this phase. This part is of around 10 to 12 years, and the entire cost will be around 1 billion US dollars. But with the help of bioinformatic tools, what we can do? We can do virtual screening. So in virtual screening, the chemical compound library is not actually a tangible library, physical library, but instead of that, all these chemical compound structures are stored in the computer itself. So we can call it a virtual library, where the chemical compound structures are there present in the database itself. And then we have this molecular docking softwares where we can see the binding activity of the protein along with its ligand molecule, means the compound from the library. Okay, so in the virtual screening, the computer software will analyze all these millions of compound. It will see how these compounds are binding with the active site of your protein, target protein, and it will find out the suitable pose, and it will also calculate the binding energy. So in the virtual screening, that software will scan these million compound and it will show you best 100 to 500 compounds which are binding with your target and which can inhibit your target. Okay, so you don't have to do that bioassay in the laboratory today. You can just do the virtual screening and you will get best 100 to 500 compounds, and from those best 500 to 100 to 500 compound which we called as a hits, we can again come down to very limited number of compound like 50 compounds which are called as the leads. So now you have to do the bioassay for only those 50 compounds or 100 compounds. There is no need to do the bioassay for millions of compounds. That was that these companies were doing in their conventional drug discovery process. Understand? So because of this knowledge based drug designing, or we can call it a computer aided drug designing, this period of drug discovery has been drastically reduced. Now you just after virtual screening, you just have to do the bioassay of those 50 to 100 compounds, and you can immediately go with the clinical trials. So what will happen? The cost for drug discovery has been reduced, as well as the the time that was spent on the discovery phase, that has also been drastically reduced. This is a very important application of bioinformatics in the field of pharmaceutical industry. Then comes forensic DNA analysis. So you must have heard about the DNA fingerprinting. Like in the forensic science, in the criminology, they use fingerprints to identify the person who was present at the site or who was present at the crime scene, because the regular fingerprints of any individual are unique. So if the fingerprint match, so we can say that the person was present at the crime scene. Similarly, the DNA profile of an individual is also unique. The DNA profile of the individual will also not be matched with the any other individual in the world. That DNA profile is called as DNA fingerprint of the individual. Okay, so you must have seen in the movies or in the crime series that whenever a biological sample is collected from the crime scene, it could be blood, it could be hairs, it could be semen. This biological sample immediately sent in the forensic laboratory for DNA fingerprinting. So in the DNA fingerprinting, what they do, they just digest the DNA into fragments and they separate it on the agar gel, and we get a binding pattern. Okay, so that binding pattern of any individual will be unique. So they match they match that DNA fingerprint from the crime scene with suspect as well as victim. So if the DNA fingerprint match with the victim, so we can say that the sample was of victim. But if the DNA fingerprint match with the suspect, then this clearly indicate that the suspect was present at the crime scene, and that leads to the further investigation. Okay, so DNA fingerprinting is a comparative method. You must have two samples to compare, right? You must have two DNA binding patterns to compare. Either you compare it with the victim's DNA or you compare it with the suspect DNA. So, how bioinformatics is useful in the forensic DNA analysis? So, in USA, there is one investigation agency called as FBI, Federal Bureau of Investigation. So, what Federal Bureau of Investigation have done? They have collected the DNA fingerprints of almost all the criminals who have been arrested, right? So you must have seen that whenever a criminal is arrested, his entire record is present with the police department or that investigation agency, with his fingerprints, with his photograph, everything. But apart from that, what FBI is doing, they are also collecting their DNA, and they are doing its DNA fingerprint. So they have DNA fingerprints of almost all the criminals who have been arrested in some cases previously. And they have stored these DNA fingerprints into the database known as CODIS. They have created a database for the storage of these DNA fingerprints of the criminals. So whenever they find out a biological sample from the crime scene, and they determine its DNA fingerprint, and if the DNA fingerprint is not matching with the victim, it is also not matching with the suspect, then what they do, they match that DNA fingerprint with the database. In the database, DNA fingerprints of criminals has been already stored, right? So they will try to match the DNA fingerprint of the biological sample which is collected from the crime scene with the CODIS database. And if they find out the match, they have the entire record of that criminal and they can easily capture him. So that is the application of this bioinformatic tools in the forensic DNA analysis. Then agricultural biotechnology. So there are many more agronomically important crop plants are there, and in these crop plants there are variety of characteristics are present. Now botany people or biotechnology people will correlate with this. You know, in plants we always try to develop some hybrid varieties with some beneficial characteristics, and for that purpose we do plant breeding experiments. So suppose I have two varieties, one is having drought resistance, another variety is having disease tolerance. So what I will do, I will just hybridize it. I will do the breeding experiment, and the progeny will have both the characteristics. Means the progeny will be drought tolerant as well as disease resistant, right? So in this breeding experiment, many of the times what happen, these hybrid varieties which are produced, they are not stable. Means the characteristics which are mixed in that hybrid variety, they do not remain stable. The hybrid variety will lose that characteristic. So we have to do this experiment for multiple generations. Now, with the help of bioinformatics, if you have a entire complete genomic DNA sequence of that crop plant, you can identify the genes which are responsible for that particular characteristic. So, if you identify the genes which are responsible for drought tolerance or disease resistance, then we can easily use those genes for the construction of recombinant plants or genetically modified plants, right? And it will also help us for efficient breeding experiments also. So that is the application in agricultural biotechnology. Then molecular phylogenetics. Molecular phylogenetics. In phylogenetics, we figure out the evolutionary relationship of the organisms. मतलब what kind of evolutionary relationship is there, which organism has been evolved earlier, which organism has evolved later, that we can see, right? We can see their evolutionary distances, we can see the divergence. So this kind of phylogenetic analysis can also be done in bioinformatics. Okay, so when this COVID was widespread in the China, maybe in the late December 2019 and early January 2020. At that time, scientists determined the genomic DNA sequence of this SARS-CoV-2, and they find out that which this virus is, and with other viruses, what is its relationship with the other viruses, how it is evolved. So for that purpose, they use these molecular phylogenetic tools, and they find out that this virus is SARS, and it belongs to the SARS family, and they find out its evolutionary relationship with the existing known SARS viruses. Okay. Then on the right hand side, you can see a picture, 3D image of a protein. Now that protein image is actually a SARS-CoV-2 spike protein structure. SARS-CoV-2, that is COVID virus, it has a spike protein, and with the help of that spike protein, it attaches to the receptor on the cells of the lungs, and it infects the lung cells. So scientists have determined this 3D structure of that spike protein, and with the help of this spike protein, companies like uh, this AstraZeneca or Moderna or Pfizer, they have developed either recombinant vaccines or mRNA vaccines. Because this protein, this spike protein, this acts as an antigen, and on this antigen there will be epitopes, right? So with the help of bioinformatic tools and technique, scientists have identified the epitope, that is antigenic determinant present on the antigen, and they developed the recombinant vaccine against this spike protein. So that's why bioinformatics is again is very useful in the vaccine technology for the development of the vaccines also. Okay, so let us move to the database section or database part. So yesterday also I told you that we need some uh, software so that we can store this DNA or protein sequence data in a structured fashion in some order. Now that requirement is fulfilled by the biological databases. So this database is a structured collection of data held in computer storage, especially one that incorporates software to make it accessible in a variety of ways. So database is a large collection of information. Now, database management is the organization and manipulation of data in the database. So whenever we submit the entry into the computer, at that time this database management will organize those entries into some ordered manner, and we can also manipulate that data in the database. That is called as database management. Then comes database management system, that is DBMS. DBMS is a software package that provide all functions required for database management, and together this database together with database management system is called as database system. So there are three different types of database systems. One is called as flat file format, other is called as relational databases, and the third one is called as object oriented databases. Okay, so flat file format is a very simple text file in which the entries are stored. Then relational databases are the databases in which the different entries are stored in the form of tables, and the interrelationship between the table is displayed. And object oriented databases are the databases in which the data is stored in the form of objects, and with the help of arrows the relation between the objects are displayed. So for relational type of databases, we can use SQL language, that is structured query language, for the construction of those databases. And for construction of this object oriented databases, we can use C++ or Java kind of programming languages for the construction of those databases. But that is a computer science part. We are not involved in that part, because once you want to develop a database, you can easily take help of the either computer engineer or the professionals who are there in the IT field. Okay. So what is the database? It is a collection of data which is structured means what? The data is not stored randomly. It is stored in some order. That is called as structured collection of data. I always explain this term by giving the analogy of our library, college library. So in the college library, the books are not stored randomly. There are cupboards or there are racks or shelves for particular subject. So there will be a cupboard for biotechnology, botany, chemistry, like that. And those books are placed in those cupboards. So this is called as a structured collection. There is some order in the placement of the book. Similarly, when the DNA sequence data or protein sequence data is stored in the database, it is stored in some order, in some structured manner. Then databases are searchable. Like when you read the book, you can search a particular topic by using an index. Similarly, database can also be searched with the help of table of contents, and databases also updated periodically, like new book versions or new book editions are updated periodically. Similarly, databases are also updated periodically, and databases are cross-reference. Cross-reference means all these databases are linked with other databases. Like maybe DNA sequence data is linked with the protein sequence data also, or protein sequence database is linked to the protein structure database also. Okay, so these databases are cross-reference with the hyperlinks. Now it include associated tools necessary for access, updating, information insertion, and information deletion also. And as I've mentioned, that data storage management is of three types: flat file, relational, and object oriented database. Now databases can be of any type, like your college must have a student database, the banks have their own customer database, right? Similarly, in the field of biology, for the storage of biological data, these biological databases are present. And there is an exponential growth of biological data, as I have mentioned yesterday also, that because of this automated DNA sequencers, next generation sequencing platforms, we are determining the this DNA sequence or genomic DNA sequence exponentially. So we need databases for the storage of that data, right? Now data could be of any type, means it could be a sequence data, it could be 3D structures of protein, it could be two-dimensional gel electrophoresis images, or MS means mass spectroscopy analysis, or DNA microarray data. All this type of data is a biological data. So this data is no longer published in a conventional manner, but directly it is submitted to the databases. Now, essential tools for biological research are provided by the biological databases, and the only way to publish massive amount of data without using all paper in the world. Now, when this DNA sequencing was started earlier, at that time, the distribution of the sequence was done by books and articles up to 1985. Then, as we started to use computer, then computer tapes were used up to 1992. Then floppy disks were used, CD-ROMs were used, FTP means file transfer protocol was used. It is still being used. Then online services were used. And with the advent of internet, today widespread use of internet is there for the distribution of sequence data since 1993. We all are relying on the internet, that is World Wide Web, for the distribution of the sequences. Now, these are some statistics about the biological databases. There are more than 1,000 different biological databases present in the world, and they are highly variable in the size. Means some of the databases are very small, less than 100 kb also, and some databases are their size is greater than 20 GB also. So there are proteins, DNA sequence databases, protein sequence databases, protein 3D structure databases, and other very smaller databases are also present. Now, out of this 1,000 different biological databases, some databases are being updated on the daily basis. Like you take an example of a gene bank, it is updated on the daily basis. Some databases are updated annually, and some databases never got updated. So all these databases, they are accessible through the web, through the internet, and some databases are free and some databases are paid. Okay. Now these are some names of the important biological databases. That's a long list. I will shorten it to few databases, and we will bifurcate into nucleotide sequence database, protein sequence database, and protein structure database. Okay. So this biological databases, it can be categorized into different category according to the type of data it store. Like it could be sequence data, it could be sequence data like DNA sequence database or protein sequence database. It could be genomic sequence database. It could be mutation or polymorphism database. I told you just that there is one database called as OMIM, Online Mendelian Inheritance in Man. So that is a mutation database or polymorphism database. Then there is a protein domain or protein family database, which we call as a secondary databases. Then there is a proteomics database in which two-dimensional gel images are stored or mass spectrometry data is stored. Then there is a 3D structure database where the three-dimensional structure of the proteins are stored. Then metabolic network databases, there the name of the database is KEGG, which contain metabolic pathway data into that database. Then regulatory network database, bibliographical database. Bibliographical means the research articles which are published or the books which are published. That data is contained in the bibliography database. Then expression databases like microarray. When you perform the microarray experiment, that data is the expression data and that is stored in the expression databases. And there are some specialized databases also there that we will discuss in detail in next few slides. So this is the this is the homepage of NCBI. Remember the full form of NCBI, it is National Center for Biotechnology Information. So this NCBI was established or it is present in the campus of National Institute of Health. And the function of NCBI is to maintain various types of databases and to host some of the computational tools like BLAST. Then this is the database at NCBI known as PubMed. So PubMed is a bibliographical database or literature database. So Medline cover the field of medicine, nursing, dentistry, veterinary medicine, public health, and preclinical sciences also. So this PubMed database contains citations, which are all the journals, all the articles which are published in the field of medicine, nursing, dentistry, veterinary medicine, public health, and preclinical sciences. All these abstracts you can find in the PubMed. Okay. Then it contains over 20 million citations since 1948, and it contains the link to biological databases and also to some journals. So new records are added in the pre-Medline on the daily basis. So this is how you can search the PubMed. We will be doing it practically also when we perform the practical. So you can see, suppose I want to find out the research articles published on mitochondria evolution. So I just go to PubMed website and in the search box, in this search box, I will type mitochondrial evolution and I will click on the search button. So what will happen? All the research articles which are published and which are having the keyword mitochondrion evolution, all those research articles will appear on the screen. So this tool is very important if you have a minor research project in your undergraduate or postgraduate. If you're doing a project, then for the literature search, you can use this PubMed database where you can find out the all articles which are published on that particular in that particular field. So I type mitochondrial evolution and click on search. You can see 3,990 results appear. Means 3,990 papers have been published which have word mitochondrion and evolution. So I can look into them one by one. I can read the abstract and I can refer to those articles if I find them useful. So this is what the PubMed database is, which is a literature database. Okay, so once you click on any of the entries, suppose I click on this first entry, energetic and genetic across the prokaryote eukaryote divide, then its abstract will appear on the screen. Now, if you want to read the full text article, there is an option on the right side. You can see full text free on Biology Direct. So you just click here, you will be redirected to the journal website where you can read the complete paper, complete research article. Okay, so this is the search box of the NCBI. You just type in the keyword in that search box and click on the go button. You will redirect it to the result page of that database. Now let us discuss in more detail about the biological databases. Now all of you have understood about the database, that database is a computerized archive in which the data is stored in a structured fashion so that the retrieval of the data will be possible by providing a piece of information which is called as making a query. Now, the databases in which biological data is stored, they are called as biological databases. Now, on the basis of content, biological databases are classified into three categories: primary database, secondary database, and specialized database. Remember the name: primary database, secondary database, and specialized databases. Now, what is the difference in between these three type of databases? So, primary database contain original biological data. Now, original biological data means the data which is synthesized by the researcher and which is not analyzed or which is not processed, which is raw in its form. So primary database, they contain this original biological data. Suppose I collect soil from my college garden and I isolate one bacteria from that soil, and then I determine a gene sequence or a protein sequence from that bacteria. Now this data, that gene sequence data or that protein sequence data, is called as the original biological data. Now this kind of data can be submitted in the primary database. Okay, so the primary database contain raw sequence or structured data submitted by the scientific community. Examples are GenBank and PDB, that is Protein Data Bank. So anybody in the scientific community can submit the data into primary database. मतलब अगर आपने कोई जीन सीक्वेंस डिटरमाइन किया है, प्रोटीन सीक्वेंस डिटरमाइन किया है, तो आप भी सबमिट कर सकते हैं प्राइमरी डेटाबेस में. Okay, without any processing. Primary databases also accept raw sequence data, raw structural data, which is not processed. Understand? The next type of database is the secondary databases. Now secondary databases, they contain computationally processed or manually curated information based on original information from the primary database. So as I have mentioned that anyone from the scientific community can submit the data in the primary database. That is not the case with the secondary database. In secondary database, we cannot submit our entries, our data. But instead of that, Instead of that, there are database curators, there are database managers who gather the data from the primary database, they computationally process it or manually process it, and then that submit that data into secondary databases. Understand? So we can say that the secondary databases contain more process data. So along with the data, there will be information also in the secondary database. Sometimes in the primary database, only sequence data is present. No information about that data is present. Then at last, third type of database is a specialized database. Now consider a situation where some scientists are working on Drosophila melanogaster. They are using Drosophila melanogaster as a model organism. Then they will not be interested in the gene sequence or protein sequence of other organisms. They will be only interested in the Drosophila melanogaster gene sequence and protein structure. So suppose we design a database, we construct a database for only Drosophila melanogaster. So that database will not be a primary or secondary, but it will be called as a specialized database because it is only focusing on a specific organism. Understand? Or another example is, suppose there is a scientific group who is working on HIV. Then they will be interested in only HIV sequences or structure of the proteins. So suppose we develop a database of HIV only, that will be also a specialized database. Understand? So specialized databases are those that cater to particular research interest. For example, Flybase. Flybase is a database of Drosophila melanogaster, or HIV sequence database, or ribosomal database project, which contain rRNA sequence data. So are the databases that specialize in a particular organism or a particular type of data. That is called as specialized databases. So I hope you understood the difference in between these three types of databases: primary, secondary, and specialized databases. We will see all these three types of databases in more detail now. So when I say primary sequence database, this primary databases, there are various examples of these primary databases. Like there is a primary nucleotide sequence database. The three popular databases are there. One is GenBank, DDBJ, that is DNA Data Bank of Japan, and EMBL, that is European Molecular Biology Laboratory. Their database name is ENA, that is European Nucleotide Archive. So all these three databases, they are nucleotide sequence database, and they belong to which category? Primary database. What are the features of primary database? I told you that they contain raw data, and anybody from the research community can submit the data into primary database. So primary database are also redundant, highly redundant. Highly redundant means same type of data is again and again repeated in the primary database. Because suppose I have determined insulin gene sequence, and other people also, they are also interested in insulin gene, so they also determine insulin gene sequence, and we all submit that insulin gene sequence into GenBank. So what GenBank will do, it will accept that sequence from all the researchers. So it results into repetition of same type of data, which we call as redundancy. So primary databases are redundant. Same data is present in a repeated manner. So the primary nucleotide sequence database examples are GenBank, DDBJ, that is DNA Data Bank of Japan, and EMBL, that is European Molecular Biology Laboratory. Then in case of protein sequences, there are databases like SwissProt, Tremble, that is TREMBL, PIR, that is Protein Information Resource, and UniProt. All these databases are protein sequence databases where protein sequences are stored. And there is only one protein structure database is present. Its name is PDB, that is Protein Data Bank. It is also a primary database which contain 3D structures of the protein. Okay. So these are examples of primary databases. Remember the features of primary database, that it accepts sequence entry or structure entry from all the entire research community. It contains raw sequence or raw structure data, and they are highly redundant. These are the features of primary database. So there are three major public sequence databases are available, which are nucleotide sequence databases, which contain, when I say nucleotide sequence database, it means it contains the sequence of DNA as well as RNA. Examples are GenBank, EMBL, that is European Molecular Biology Laboratory database, and DNA Data Bank of Japan, that is DDBJ. All these three databases are freely available. You can access them free of cost. The small number of sequences, especially those published in 1980s, were entered manually from the published literature by database management staff. But now it is precondition. If you want to publish a sequence into article, then it is precondition that you have to submit that sequence to any one of these database. Then and then only your paper will be published. Otherwise not. Presently, sequence submission is a precondition for publication. Then all these three databases, that is GenBank, EMBL, and DDBJ, they collaborate and exchange new data on the daily basis because they have established collaboration among themselves. The collaboration name is International Nucleotide Sequence Database Collaboration. This INSDC, that is International Nucleotide Sequence Database Collaboration, has been done in between GenBank, EMBL, and DDBJ. And according to this collaboration, they have agreed that they will share data with each other. They will exchange data with each other. So even if you submit your sequence entry to GenBank, overnight what will happen? It will share that entry with the DDBJ also and EMBL also. If you submit your entry, means if you submit your sequence to EMBL, then it will be exchanged with DDBJ also and GenBank also. That is what happened because of this collaboration. So although the three databases all contain the same set of raw data, each of the individual database has a slightly different kind of format to represent the data. So file formats of these three databases are different. Now this is the collaboration that I mentioned about, the International Sequence Database Collaboration. Now you can see this GenBank is exchanging its data with EMBL, it is exchanging it with the DDBJ and GenBank, and all these three databases contain the same type of data because they are exchanging the information on the daily basis. Now GenBank is maintained by NCBI, that is National Center for Biotechnology Information. Okay, National Center for Biotechnology Information. Now all the databases of NCBI can be searched with the help of a search engine known as Entrées. Do not pronounce it as a Entrées. The proper pronunciation of this is Entrées. Entrées is a search engine for searching NCBI databases. Okay, so new sequences you can submit into NCBI, you can update your existing sequences. Then there is another database called as EMBL, European Molecular Biology Laboratory. This database is maintained by EBI, European Bioinformatics Institute, and you can search EBI databases with the SRS, Sequence Retrieval System, SRS, Sequence Retrieval System. Okay. Then DDBJ, that is DNA Data Bank of Japan. Now this is maintained by the CIB, that is which is a part of NIG, National Institute of Genetics, and you can access this CIB databases with the JetEntry search engine. Okay, so these are three earlier to collaboration. Before this collaboration, all these three databases were independent, but after collaboration, the data present in these three databases is same. Now that was about the nucleotide sequence database. For the protein structure database, fortunately, we just have one database known as PDB, that is Protein Data Bank. Okay. Okay. So as I have mentioned that in case of nucleotide sequence databases, there are three databases: GenBank, EMBL, and DDBJ. These are most popular databases. But for protein structure, there is only one database. The name of that database is PDB, Protein Data Bank. Now, this Protein Data Bank, today it contains more than 2 lakh protein structures which have been determined experimentally. So, there are three experimental techniques for the protein three-dimensional structure determination. The first one is X-ray crystallography. Then second one is NMR, that is nuclear magnetic resonance. And there is cryo-electron microscopy. So cryogenic electron microscopy. So these are the experimental techniques through which we can determine the protein structure. And when we say that we have solved the protein structure, it means that you have atomic coordinates, three-dimensional atomic coordinates of all the atoms present in the protein. Understand? So this information of 3D coordinates of all atoms of the protein, they are stored in the PDB database. Once you determine the structure by X-ray crystallography or NMR or electron microscopy, you have to submit that structure to the PDB if you want to publish that data. So you can find out the protein structures which are which have been determined earlier. You can simply visit the PDB website and you can search for the specific protein structure. Then comes the secondary databases. Now, secondary databases, they are different from the primary databases in the way that anyone cannot submit the entry into secondary database. The secondary database curator or operators or managers, they collect the data from the primary database, they remove the errors from that sequence entry, and then they annotate that sequence, and then it is submitted into the secondary database. Okay. So what is the sequence annotation? Sequence annotation is simply the description about that sequence. Description about that sequence. For example, suppose I have a protein sequence. Now in that protein sequence, no information is given, means that protein sequence has not been processed, then you will not get any kind of knowledge from that protein sequence. But if you process the sequence, if you analyze that sequence and you find out what is the function of that protein in the cell, what is the subcellular location of that protein in the cell, or what kind of post-translational modifications are occurring in that protein after its translation, right? What kind of cofactors bound to that protein. If you find out all this information and you associate this information with the protein sequence, the sequence will become annotated sequence. Understand? So annotated sequence and unannotated sequence, difference is only of information. In annotated sequence, along with the sequence, there is information. And in unannotated sequence means in the raw sequence, only sequence is present, no information is given. So primary databases, sometimes primary database contain this unannotated raw sequences, but nowadays many of the primary databases also become annotated. But secondary databases are compulsory annotated. They contain annotated sequence information. So to turn the raw sequence information into more sophisticated biological knowledge, much post-processing of the sequence information is needed. So there is example like uh, Uniprot knowledge base. Now in this knowledge base database, the annotated sequence information is present. And apart from that, Apart from that, there are databases like prints, prosite, pfam, prodome. All these databases are secondary databases which contain information such as protein domain, protein structure, protein family assignment. All this information are present in the protein secondary database. So the annotation of each entry is carefully curated by human expert, and thus it is of good quality. The protein annotation include function, domain structure, catalytic state, cofactor binding, post-translational modifications, metabolic pathway information, disease association, and similarity with the other sequences. So you can see these are the examples of secondary databases: ProCite, PFAM, blocks, prints, prodome, which contain protein domain information, protein family information, like that. And finally, we have to discuss about the annotation in greater detail, because secondary databases contain annotated information, more annotated information. So annotation include open reading frame, functional sites, structure and function, like that. So you can see on the screen, this is the gene sequence. But by just reading this gene sequence, you are not getting any information. Can you tell me which gene sequence it is? What is its function in the cell? Which protein is coded by this gene? Where the promoter is present? Where the open reading frames are? Where the transcription termination site is present? And by just reading this sequence, you will not get any answer. In order to get answer, you have to process this sequence by using some software. So once you analyze this sequence by using computational tools or by using existing literature, you can convert this sequence into annotated sequence. This is a raw sequence. On the screen, whatever you see, it is a raw sequence. After processing, it will become annotated because you will add description to the sequence. See, now this is the annotated sequence. You have identified the promoter site, then transcription factor binding site. Okay. Then this is the ribosomal binding site. Then in blue color, you can see they are open reading frame of the coding sequence, like that. This A particularly, this one is a transcription start site. So once you analyze that raw sequence, it will convert into the annotated sequence. In the annotated sequence, along with the sequence, information is also present. Detailed information is also present. Now, how will you come to know about this promoter, open reading frame, transcription factor binding site? So there are software to do that. You just submit your gene sequence entry into that software, and that software will predict that where these particular sites are present. And you just have to confirm it in the literature, and you add that annotation into your sequence.